one, and don't laugh at me. And you're doing it already. And we are recording with Miss Martha Fowler, who was on last week with Dr. Aditi Bhargava. And as last week, and as Dr. Bhargava says with UCSF, and Dr. McCullough says with Baylor, you with Rice University, nothing you're saying represents them. This is you speaking as yourself as an individual. And nothing I say represents any of these institutions or anyone I've ever interviewed. But before we go further, if you want to kind of elaborate on that disclaimer to save yourself, take it away.
Yeah. Thank you, Tommy, for having me on your podcast again. I appreciate it. And I'm excited for us to have our conversation. Yeah. So again, anything I say does not represent any of my affiliated institutions. This is purely based on previous knowledge and my own independent research.
So I did read your entire thesis. For everyone listening, I saw the email at 3. I started reading at 6. I did go through 73 pages. I don't know how much I retained, probably next to nothing, but it was a point of pride for me to get through it all. So let me say it in my dumbest possible way before you kind of go into what it actually is. The core of them regenerate, and I'm not going to use any medical terms, so you're going to know what I'm talking about. But for all the people listening with me, the core of the tumors or the neurospheres regenerate themselves. Those are like a 3D printer. I use really weird analogies, or just a printer. The outside are kind of like what has been copied. So let's say you are spreading flyers or something. Pretend you're spreading misinformation flyers. The interior is the printer. It has the ability to print other printers, self-regenerating, the progenitor cells. And it also has the ability to do whatever the extraneous thing is, the movement, whether it be the oligodendrocytes, the axon, myelin sheathing, all along the white matter tracks. That's putting up the flyers, right? It's putting up the flyers throughout the city, and people are trying to tear them down. They're like, take it down, take it down, take it down, like the angry dictator. And there's kind of 2 ways to go about it. One mode of attack is we have to take down all the flyers, but the printers are still there. The other mode is we can take down the printer and we'll have tumor regression, meaning that there aren't any new flyers, but there are still the existing flyers out there. If you just pluck the flyers, the printers can regenerate themselves. And with that, it also doesn't move chaotically through the mass of the city. It moves along tracks, the roads, the sidewalks, like electricity going through the air. It's going to find the metal. The heat is going to find the copper, like in a cooling system. It's going to go through along those lines of least resistance, be it electrical or in this case, biological, the scaffolding. It goes along those lines, and that also allows it to evade things. So if you find that cancerous piece, you got to go in with a scalpel, or it's not 1910 anymore, whatever femto lasers they're using to remove those bits. But you literally have to get, and no one uses that word correctly anymore, but you literally have to get 100% of them, or the self-regenerating the progenitors in the core of the neurosphere, the SOX2 and the OLEG2 rich transcription factors, which you use with the immunofluorescence to find them. I do remember that from biology. They will still move along. They can regrow themselves. And it also seems you have the go or the grow hypotheses. So if you're the kind of and there's another thing I thought of. I just read a book, as I was telling you, the most recent books are always where I use analogies from. I just interviewed Richard Rhodes last week for a book about ants. It was actually really interesting. But they would move in this mass, and sometimes they would move in such a mass that like the queen and like a bunch of food, and I'm talking like 10 million ants at once, not like a little ant pile. It's creepy. I mean, like 10 feet wide, like you can actually hear it. That's how many ants there were. But in the center would be like the queen, and there'd actually be like food morsels or something. And along the outsides, you'd have these little nodes, and I'm going to forget the word, but whatever the thing is for the moving, was it palette, something with a P, about like moving nodes of cells?
Yeah.
Those are the ants on the exterior. Those are more rich in OLEG2 because they are always constantly growing. The ones in the center, the fat queen bad bitch who's regenerating, you would say more queens, which doesn't really work with ants. That's in the center that has less of those, but it always has to be growing. If it's not growing, you got to get to the exterior and start moving out. Mom and dad kicking the sun out. Hey, you're 30 years old. Get out of the house. And also, you have to keep doing that because they can't grow forever, because you have, I can never pronounce it, apoptosis, where the cell death, cell suicide, because the bigger the mass gets, kind of like a planet can only get so large before it collapses in on itself and undergoes fusion and becomes a star, the center doesn't get nutrients. So it actually doesn't want to be perpetually growing outwards. It's grow or go, moving along. So you make your own scaffolding with your at-home contraption, which you said wasn't as good as the commercial ones, but it's still kind of badass. That was some Tony Stark shit, like making it in a cave. You can make that with I remember with your M electrode and everything.
Yeah.
You make that.
Not for my name at all. It was just it wasn't. I wasn't trying
nothing to do with
it.
it. There wasn't an F1 shape. There has nothing to do with it's. You have those, and they move along the tracks, and they maintain their stemness, and so a lot of it, and I say this very relatively, I feel like a lot of it was probably not much of a surprise. It's like, okay, it moves along the tracks instead of in like the chaotic middle ground. Just like I drive my car on the road, I don't just drive through apartments to get to Walmart, right? It kind of makes path of least resistance, sure. Maintaining the stemness seems to be how you treat it, because it can move along these tracks. It stays within the nervous system, doesn't go out to other stuff. So it clearly has an affinity for whatever myelination thing's going on. It maintains the stemness. The ants are always coming off of the main mass. The queen's always in the center, regenerating itself. And even if it stops regenerating itself, it can actually take some of the other ones that have been differentiated and bring them back into pluripotent stem cell or quiescence. So something to do with being on the tract maintains their stemness, which means they can go dormant, and then even after surgery and you blast it with everything, they can kind of poke their head up and be like, all right, we're good. And they can start growing again. So the trick comes down to somehow, if I think, attacking the core of the neurosphere to stop the growth, and something to do with the transcription factors, OLEG2 and SOX2, which aren't only on those, they're on everything. They're just in different parameters here. You want to attack those to avoid or to at least slow down growth, because like you said, even with maximal treatment, it's 12 to 18 months. And because you either have regression or you have like momentary pausing and then relapse, your idea is to kind of do like a two-pronged approach, much like the M. Well, I guess there's three-pronged approach. That doesn't work to get it. And I know I just ranted for like 10 minutes. As someone who has not taken a biology class in almost a decade, how'd I do?
That was really good.
All right,
really good.
Podcast over.
Yeah, we're done.
All right. Your thoughts on that? How do you explain it more intelligently?
Yeah, that was spot on. So the main thing that we were having trouble with was actually, so we had a home built electrospinner is what we call it. So essentially that's, you apply an electric current to a solution, like a polymer solution in a needle, and then to a target that would collect that solution. And that electric difference, or that difference in electric field, is actually going to cause the solvent to evaporate. So you're left over with whatever polymer you put into that solution. That's going to make the scaffold. And then you can design the collector to dictate how that scaffold is going to turn out in its morphology. So if you just had a flat aluminum collector, the fibers kind of just fall on there randomly, and you can change the parameters when you're electrospinning, like the flow rate, the electric current, and that would all dictate how those fibers would look, like how large they would be, how porous the scaffold would be. So where we ran into troubles was actually trying to create aligned fibers. So it's actually something that can be pretty problematic if you don't have a commercial electrospinner, a high grade electrospinner. So what I then went ahead and did was try to invent or come up with some kind of design where you can build this system, right, in your lab without purchasing a fancy one, and you can get aligned fibers. So that's where I came up with these different targets, and it's actually kind of funny because I the reason why I guess I got interested in bioengineering was because of this project, because I would go to Home Depot and just spend hours there looking at parts, like looking at different pieces and trying to come up with designs and what I could fabricate. So I would just go in and buy a bunch of aluminum, just a bunch of tin and all that, I would just go in there and bring it all back to the lab and think what I could create. I remember for one trip to my parents' house with my stepdad, we just sat at the dinner table at all these pieces and we were assembling it, placing it onto like this wood pallet to create all these designs. So it was a lot of fun. But then I had to test each one of those in the lab and try and encourage alignment of these fibers, because I wanted to really study brain cancer. And so this was one problem when I was doing my research that seemed to be the most interesting. We already had the electrospinner in the lab. We could already design random fibers for wound healing, actually, is what we were applying them to create dermal wound mimics. But then I used that technology and found some kind of collector that would create aligned fibers, so in that way I could study something in the cancer field. So brain tumors more specifically. So yeah, and then from there, I developed collaborations with a lab that had patient-derived brain tumor samples. Glioblastoma is what they're called. And what was really different about these samples is that they were enriched for the stem cell population. So at that time, and even now in the literature, one of the things that's responsible for patient relapse is that stem cell population in that tumor. So what's interesting I almost think of tumors as its own little organ, because every organ in our body has its own cells, right? To replace it.
Vasculature, movement,
Yeah. So it's interesting. It's tumors are almost kind of alien when we think of them, because they take the best components, right, of our body. They can create vasculature, right? They can get their own, it's called angiogenesis. They can get their own blood supply to them.
They're creepy, dude.
They use up, a lot of sugar and glucose. So they use up a lot of energy for our bodies. And then they have this ability to metastasize and move wherever they want to in the body. So yeah, I got really interested in the stem cell component because when I was just doing my, so whenever you are designing a thesis, you look into the literature, lots and lots of different articles, and try to find what interests you and where a gap might be. So I was more interested in the stem cell component because it was the component that has been linked to chemotherapy resistance and patient relapse. So creating these or growing these neurospheres then in the lab, when you grow them in a neurosphere culture, it actually helps maintain their stemness, so we call it. That's a funny word, but so it helps retain, because when cells actually go onto a surface, they'll actually start to, we call it differentiate. So they'll actually start to change their phenotype. So that's why we have to grow them in suspension cultures. And then once I was able to create aligned scaffolds, then these spheroids were placed onto aligned versus these random scaffolds. And I really wanted to focus on, again, it was just a master's degree, so it was a bit short. I would have wanted to do a lot more in the stem cell component. So I wanted to look more into like their resistance, and especially with those markers. So when I wanted to show that the cells, when they migrate, even though they attach to a surface, because usually, right, when cells attach to a surface, you'll see differentiation. So I wanted to show, right, yeah, so I wanted to show that these cells, even though they're moving throughout a brain, I wanted to create a mimic where you could show that the stem cells could move out of a tumor, right? And that they were going to be the ones responsible for moving out versus like the differentiated cells. And then because if you have the stem cells that are mostly moving out and away from the core, and those ones are responsible for, hiding away from the resection, right? So when they resect the large tumor, you're going to have some residual cells.
Sure.
So some of those
Residual.
residual cells can be stem cells, right? So if you have stem cells left over after resection, those can then give rise to a whole new tumor because they will start to divide. So they'll give off another stem cell when they divide, and then another cell that will probably go towards differentiation. So it's going to encourage another tumor to form, and that's how patients relapse. And then these stem cells are chemotherapy resistant. So TMZ was the big chemotherapy agent for glioblastoma, and it's been shown that they have a resistance to that chemotherapy agent. So it was a pretty fun and project, and I like when there's, that's usually the case with science, right? We don't have all the answers, so you can investigate anything and find a gap.
Yeah, no one can say you're doing it wrong because you're like, well, you don't know what's right. So technically, none of us do. Yeah, I think you could almost make like a loose analogy to like early life. You could say, and this is going to be really loose, and I'm making it up as I go, so excuse me. Like early life, like single cellular, maybe multicellular, like throughout the ocean, and that's not a. As different life forms reach different areas of Earth, granted it's over billions of years, not in the lifetime of a cell or something, but you can sort of see if you do like a time lapse of all of like life, you could see as, like you said, it hits the surface and then it starts differentiating. The genotype switches. Hey, turn that off. Now we need more of this because we're on the ground or we're near a light. You can see that life takes different ways. I mean, we know that there's not just cells, but I mean, we know that there's life at the bottom of the ocean. We know that there are, birds that can fly for an insanely long time. We know that there were once dinosaurs. We know that we have us walking around, slowly becoming more and more hairless, standing up. We don't have the coccyx anymore. The brain's moving more, opposable thumbs. And then there's still other things where something like the Galapagos Islands, right, where you get genetic drift. You get something over here, adapts, no more or less different than a penguin adapting to Antarctica or a camel adapting to an African desert. You get them on an island and then they can start to drift within there. That seems to be more like the Oleg 2 kind of the quote unquote movement. And I know this is a very different analogy because we're talking about cells reproducing in a 40-hour trial over, 4 billion years, but versus you could say like the stem would maybe be like, again, it's not really working, this analogy, temporally would be that original single cell or multicellular thing where it's almost like if you took that and somehow put it on like Mars and it didn't die, it would take a long time, but you would eventually get a biosphere. But you could say the stemness, right, that central part is like the original kind of seed. And the problem is it's not that you just find that and go, well, there it is. Well, everything that has Oleg 2 and SOX 2, we just kill. And it's like, well, funny thing about that is like kind of everything else in the brain, the very thing we're trying to save. Oh, there's, I've told you
the expressions just increase, right? So you have the
yeah,
expression
70
differences.
to 90. Yeah.
Right. But that's why, right, you wouldn't want to essentially target that necessarily, right? Because all your other cells have that,
Yeah, because you couldn't, it's the same thing. So I was thinking as you said this, well, one, it also kind of sounds like the problem with to do a complete 180 carbon nanotubes. This isn't in alignment with all the microscopic stuff, and nanoscopic stuff isn't always about alignment, like the difference between just like graphene and so that's insane that you were able to do that. But I was thinking of another thing. There was I think they did it in Japan. They did this study where they're trying to find the most efficient way to lay out a new subway system. I don't mean like construction methods. I mean like if we're going to build the subway, what is the most efficient? Do we want a station here, the movement of people? Just a trillion factors, right? The habits of people, the businesses and everything, the culture. And they came up with like a design. It looked pretty good. And so what they did was they're like, well, hold on, let's look at like the United States. And I think they chose like the Eisenhower interstate system between all the big cities. And they like mapped it out, kind of like you did with the graphic design, which I did understand graphic design, going in with the TIFF image and moving all this stuff and using the freehand. That part I got that. I got that from making memes. But what they did is they showed it as, seriously though, they looked at, they're like, here's the United States with all these different hubs: San Francisco, Texas, or Houston, Boston, New York, LA, whatever. They then wanted to see what would happen if you took a life form. And I think they used, I don't know why they used psilocybin, the psychedelic mushroom, but they did use that cubensis mushroom. And they put the spores on some agar. And they wanted to see how would it, because it will do it in the most efficient way, much like it'll just, like a force, it'll use all the nodes going out and sending back feedback. Don't go that way, go more this way.
Yeah.
And it actually did kind of develop its own quote unquote subway system that looked pretty close to ours, but it was like 30% more fit, which is everything when you're talking about billions of passengers. It looked pretty close to ours, but there were some things that they were like, oh, we never even thought about that. The point of that whole rant is it really comes down to like a question of two ways. Is the cancer just perpetuating itself and just kind of growing for growth's sake, and it found that going along the white tracks is the way to go? Or are there different parts of the brain, because you said it stays within the brain, so it's not, it would just kind of, I guess, go to your fat or something if it just wanted nutrients all day long. Could it be possible that, let me take it back to the agar analogy, they put little like nutrient sources where the cities were? Are the cancer cells, are they moving towards parts of the brain that maybe we're looking at as just haphazard, unlimited growth? Could it be possible, and I don't even know if this is within your specialty, could it be possible that there are parts of the brain that it is maybe trying to grow towards? Are there more nutrient, and I know that sounds stupid, but like, is there a method to their growth? Or there clearly is. They're not going through the random gray matter, they're going along the white track matter, and they're pretty good, they're pretty sneaky bastards, like they figured it out really, really well. And then it's a catch-22, because then it kills the brain and then the whole thing doesn't work anymore. But if you could run that forever, which is what you're doing with your scaffolding mimicking it, is it possible to, if you could just run that simulation longer, what would be the long term? Could you see, is it actually trying to make almost like some meta structure? And if you could visualize that meta structure, would that be a way to fight it? Like the forest fire isn't random. I think it's going to the other parts of the forest. Like maybe if we cut it off, is that anywhere near reality? Or have I gone full just off the deep end?
No, I feel like you're asking good research questions.
Well, I did research with fish.
Think you're you could go get your PhD.
Screw that. Not for me, dude. I gotta yell in front of a camera.
But no, I mean, but that's what I think research takes: creativity, right? A creative brain. Because you got to think like that, right? Think outside the box. So to answer your question, yeah, I don't know 100% the answer to that, of if there's a kind of destined movement. I think again, like we're saying, they're going to take the path of least resistance. I think too, they're going to take the path of survival. So they also excrete something called exosomes. So they actually communicate with each other through the material that they push out of the cell. Each cell can communicate. So when we actually did some of the microscopy on the scan electron microscopy on these cells, within one individual cell, you can actually see all the small little exosomes, and another cell nearby with the exosomes along the fibers. So, but that's a whole other field, that like even that communication, like someone could do a whole PhD
Yeah.
thesis just on that, and I'm sure it's already out there somewhere. But yeah, so it's very interesting. We don't know if there is a method to the madness. I think what I would hypothesize is that, yeah, they're trying to survive, and right now when they adapt to that environment that they're in, they're going to change the way they're going to change because they're going to get used to that environment. So like there's a whole thing of when a cell goes, let's say, from the skin into the bloodstream, it's called epithelial mesenchymal transition. So they actually change their phenotype, and they're changing to survive in another compartment, right, is the way I like to think of it. So I mean, they're very adaptable. It's if our organs were all that way and tried to survive all the time and live forever, yeah, that'd be great. But it seems like these tumor cells can do that, but then the catch, right, is that it's not functional.
Kills the host. It's not.
Yeah.
It's not long term. It's not.
Yeah.
Yeah.
I mean, because the tumor, right, can outlive the patient
Sure.
sometimes, right? I mean, we all have mutated cells that our bodies can detect and can get rid of. But in the chance that a tumor does occur, tumor cells, I don't know if you know this, but they have, do you know what telomeres are?
Yeah, the shortening of isn't the length of your telomeres, doesn't that depend on like your life? And that Ray Kurzweil is
Yeah.
going into that with his book, and like, can we lengthen the telomeres? Wasn't there like some supplement people were pushing? I think it was like lion's
Yes.
mane mushroom.
Telomerase. So the enzyme that extends it.
Yeah.
Telomerase. Yeah.
Yeah. There's I think it was lion's mane mushroom, and it was like a supplement you could buy, and it had like the only like naturally occurring like like nerve growth factor, and they thought that has something to do with telomeres. I don't know how far that went. Sorry.
No, it was a whole field, actually a
Yeah.
therapeutic field, right? They were actually thinking that this is how you could live forever.
Yeah,
but you turn into essentially one non-functioning blob, right? Because it's
but you'd live forever.
Parts of you will, right? Essentially, that's a tumor, right? It's part of you because your genetics and
that's some horror movie shit.
It lives, yeah, right. It really is. But so tumors can do that, right? Tumors can extend their telomeres so that's why they're called the immortal cell, pretty much, and we can extend them and grow them, however we want, and they will actually signal telomerase, and that goes back to more of that. What's interesting about tumor is that you can think of cancer research also involved or similar to developmental biology, right? Because you have these stem cells that are behaving a certain way. For example, like in developmental biology or in the fetus, right? Your DNA does actually have to be long, right? So telomeres or telomerase is going to be there, but once you're born and all that, your DNA, it starts dividing your telomeres, and every time your cell divides, that telomere gets shorter, right?
Mm-hmm.
Nothing extends it. So tumors, in a way, just take on the form of, at least that's the way I think of it, of something that's first developing, right, at the initial stages of life.
Okay, yeah, like early like physiology. I mean, like embryonic. Yeah. Okay.
Yeah, and it needs, like I said, it needs a lot of nutrients to grow and divide. And when they first discovered the angiogenesis, actually, too, whenever they, you discover a component of the tumor that could be contributing to its survival, you try to cut it off and see if that's going to be a treatment, essentially. So the person that found out about angiogenesis, right, that was going to be the cure all, like, we found a way to cut off their nutrient supply, right? Because when you think of, okay, if you cut off nutrients to something, cut off the food source, it should die out, right? Okay. So an it's called VEGF is what they secrete to create these new vessels. And so they thought though that if you did like an anti-VEGF treatment, that right, they wouldn't be able to allow angiogenesis. So they would. So then when you think about a tumor cells that are clustered without nutrients in the middle, it's actually going to be necrosis. It's going to
Uh-huh.
die, right? That's like when we can't get oxygen, right,
Yeah.
to a certain part, right? It becomes necrosis. So same thing with a tumor, it becomes necrotic in the center. But the issue with that is that we know that these cells can migrate and degrade matrix even around them. So they found all these other things
Jesus.
that the tumor can just
Just this
escape.
cascading death machine. Like if you kill it,
Yeah,
it's wearing a suicide vest, and it's also going to blow up everyone around it. Is
that's why
there?
there's so much to cancer research. I mean, you're right. I mean, you think, they thought like, oh, angiogenesis.
Oh, it's
Let's
done.
just,
We did it. We did it,
yeah, we're
guys.
good.
We did it. We found it
Yeah.
out. Penicillin. That's the end of medicine. No. Now, is there a way that you could, instead of cutting it off, could you, I guess, two modes, and you're the researcher, not me, so please cut me off if I'm being stupid. Could you either just send it like a false signal and say, oh, you already got all the nutrients, man, you're full. Like you're full. Or could you almost sort of like judo it, where it's like you don't try to stop the punch. You like, what is it like? What's the Sun Tzu stuff? You grab the punch and move forward. Like you use their force against them. Is there almost any way you could like I don't even, and this shows the limits of my medical understanding. Is there almost a way you could do what we were talking about earlier? Could you almost like put in like the least lethal part? I guess I would imagine, I don't know anything about neuroscience, but I would imagine there's parts of the brain where it's like, if you get a tumor there, it's like, dude, that's lights out. Versus if you get a tumor, I would rather break a finger than a spine. There's probably parts of the brain where it's like, if you got a tumor here, like, hey, this is a good spot to have it, like all things considered. I wonder if you could almost put in nutrients or the chemicals that indicate a nutrient rich mass. I wonder if you could put that in like a less than lethal part of the brain. Like, yeah, just kind of like leading everyone over here, like, hey, follow me. Like,
Right.
I wonder if you could do something like that. So instead of just trying to brute force stop it, kill it, cut off the vascularization, or if instead it was just like, putting treats down so the dog follows you down the hall, and then like you kick it outside and you're like, go take a shit. Like you could, versus just trying to grab the dog and it fighting you. I wonder if you could sort of almost like art of war, like lead it along, but then would it just keep growing and growing, and would it start to be like, oh, we've got nutrients everywhere, and would that just make it worse? Not that you know the answer, I certainly don't, but are there ways to go about it like that, or am I just not even close to the mark?
Yeah, no. I feel like that's a good idea. I'm sure there's probably somebody
Yeah.
out there that's investigating that, because like I said, exosomes, for example, is how you
Yeah,
signal,
yeah,
right?
the communications.
Yeah, so I mean, there could possibly be a way that you could do some kind of communication to recruit, right? All the cells to a certain area, um, or try and recruit. But I think, it's so hard to know where these cells are going. We know of certain things of, okay, well, they get into the lymph nodes, all of that. But even we, don't fully, I mean, there's all kinds of hypotheses on how it actually gets to that points and the steps it takes and the role that the nutrients play, what enzymes are being secreted, and how your immune system is even reacting. And that, I mean, even so, understanding the distribution of the small individual cancer cells, along with, they use the term clearance. So when they leave an organ and can enter the lymphatics to then be, in an area where your immune system can get triggered and hopefully detect them. But we, sometimes we don't even understand that step process. And so that's why I like tissue engineering, because you can pretty much make like a little benchtop model
Yeah.
to look at those signals, right? Because in a human, you can't go and try and figure out those signals, with our imaging technology is good, but you can't see down to the molecular level, right?
Excuse me, aspirin.
Like,
So what, but to go on that note, so what I was going to say is like, everything's kind of dependent on the model, right? If we for sure we know the earth is flat, so let's just work from there. We're working on everything. It essentially, or eventually someone asks like, hey, how sure are we that the earth is flat? Because like all the work we're doing is dependent on that. Well, it's like, what do you mean it's flat, dude? And when you find out it's not, it's like, well, for the most part, nothing's different. Like gravity is still the same. Like if you want to move in that direction, it's like you have to have an opposite force. But for much bigger things like orbital mechanics or over the horizon sight, your model falls down if you're looking at a sheet versus a actual sphere. And I know that may upset some people. Somebody emailed me last week asking me if I wanted to know the truth about the flat earth, and I sent them the episode I did with Charlie Duke, who walked on the moon, and said, sorry, this guy debunked it already. I'm sorry, but the model matters. So could it be incorrect? As like you said, there are limitations to your homemade Tony Stark Iron Man model versus a large commercial. But even the large commercial model, as you were saying, with the exosomes and the transcription factors, and hey, we got it figured out with angiogenesis, and then you go down decades later and you go, dude, we had no idea what we're even talking about. Could it be beneficial that the model that you're using, right? You talked about like gelatin is this, we can't use that, we can use hyaluronic acid or all this. I'm butchering most of them. We're using these to create these models that, right? And you're talking about the different ways to get the strands with the M electrode. And do we want this kind of fibrous thing? Do we want this kind of whatever sort of mass and fractal scales of what would represent the white tracks in the brain? If the model isn't 100% correct, because you're talking about the diameters of stuff and how much does it differentiate, and although I failed calculus 3 times, I can only imagine that there's probably some equation that explains how much it breaks off, like highways and off ramps and cul-de-sacs and sidewalks. And is there a silver lining in that the model might not be entirely accurate? It would be arrogant to assume that it was accurate. We can't get anything. Our supercomputers can barely model folding proteins, right, down to the atomic scale. If the model isn't wildly accurate, could there be an answer to the question that maybe lies in, it all seems good, hey, the water bottle still falls at 9.81 meters per second on our flat earth, versus, hey guys, but if we want to shoot the water bottle over the horizon, we can do it, but now it's a spherical. Is there something there? And is that kind of like a worthless question? Because it's like, yeah, with more accurate models, everything would be better, Tommy. Congratulations. Like, it's not a very, it's not a very new thought. Is there anything in there? Like, could there just be like that sort of eureka moment? Could it be in a more accurate model? And if your research shows that this is viable, well, then couldn't you do that working for like a big farm? And I know that they kind of have a bad rap right now, but couldn't that work with, hey, forget this homemade stuff. Do you want to use like an actual mouse with fMRIs? Like, what if you had, does that make sense? So 2 questions. Is the model accurate? Is the model accurate? And is it close enough that there aren't any secrets hiding in the shadows? And if this was shown to be commercially viable, would that sort of open the floodgates of funding? If you get the proverbial cure to brain cancer, that holy grail, would those things, not having to use this homemade stuff, but actual like tissue models, actual stem cell lines, if you just had, and again, not a good rap right now, but if you had like a Pfizer just opening the pockets of the purse strings. So those 2 questions: is the model accurate, secrets in the shadows, and what would unlimited funding do?
Yeah, so of course, so I think of it as a tissue engineer, you have to, or tissue engineering training, let me say that way, not fully tissue engineer yet. But when you are doing this kind of research, of course, you have to take the materials into question. So we call, they're called biomaterials. So materials that would be compatible with the body or compatible with a living system that's not toxic, right? So at first you have to know, right, that your material is not going to be toxic. So this, whatever you're seeing isn't, right? If you're like, oh, the cells are dying on the line. No, it's just that they're toxic, right? That material is toxic.
You're using
right?
arsenic. Yeah.
Yeah. Right.
It's like, no, aluminum, not arsenic. my plutonium foil isn't working. Well, Martha, get tin foil, not plutonium foil. Yeah.
So, I mean, you have to do experiments in between there to show, right, your material is good, your, you have to take into consideration even things like hydrophilic versus hydrophobic. And this goes across, biomaterial platforms for anything, right? And I know we had this discussion in the last episode, right, about nanoparticles. Right, the same thing can be applied to biomaterials just for, developing some benchtop model to understand a molecular process that we couldn't understand, normally in a human model. So what you would do then to that, I'm not going to say validate, but to check if your signal or what you're seeing is accurate in your benchtop model is then put that in a living model. So like a marine model, mirroring. So that's a way to check. So you would look at, okay, now I'm going to have a model where this signaling or a genetically modified one where, this signaling is down, right? I'm going to put these cells, maybe that are expressing this protein in there and see, if I can see the same thing, or if you take the cancer cells and put them into a we call them in vivo model, and you see that they're behaving the same way that they did on a mimic. They're choosing the aligned fibers, right? That would be one way to validate or to check if it's accurate, right? But of course, there's going to be things, because the way I see it, right, cells don't evolve in a plastic environment, right? They evolve in humans, right? Where they have proteins, they're surrounded by fluids, right? It's a very diverse environment that they're from, but we're isolating pieces of that environment to see what could play a role, right? But you're not going to be able to create a whole person, right? On a benchtop. So the goal is to try and, there's all kinds of cool things now with microfluidics. Like I'm learning how to 3D print, which is so fun. So you learn, there's all these different tools that you can now apply to a system to try and get it closer to what we might see in the body. So if I want to see fluid dynamics now within the brain and how that might influence, across white matter tracks that these cells would see, maybe that would change, right? Because the only thing I was looking at were white matter tracks, but there's a million other things, right, going on, right? There's electrical, there's electricity, right? Environment. So I chose a hydrophobic polymer because your myelin sheaths are also hydrophobic. So we thought it was interesting that the cells were designed to grow across something that was more hydrophobic in nature, and that they were choosing these myelin sheaths. But it could have more to do with alignment rather than the hydrophobicity. So that's, another interesting question. But yeah, so I think that there's gonna, it's gonna take time for it to get up there. Things are progressing pretty quickly, but even so, a lot of people, when you get into tissue engineering, will say what are the ups and downs between electrospinning and 3D printing, right? Because each technique will create something different, right? So you can also combine techniques if you wanted to as well. You can also 3D print with cells in a liquid, right? So you can print cells into a construct. There's all kinds of experiments going on of printing, a bone construct, right? People are seeing that, as the future. But you have to put, like for a bone, you'd have to put calcium phosphate, right? Is one thing to consider. So you gotta actually consider the materials that the cell is encountering. You have to consider the actual architecture as well as the materials. So yeah, there can be quite a bit, and it can, there's a lot more work, of course, that needs to be done, I think, in the field before we're like, oh, this is going to be the one thing that gives us answers.
Yeah, it's like,
that's how I feel with everything. I think it takes time.
Right. Warren Buffett, you can't have a baby in a month by getting 9 women pregnant tonight. It takes 9 months. Some things just take time, regardless of the effort or the intensity. My older brother is it took like, I don't know, Spanish in high school or college, and he for several times now he's kind of actually like lived in like a like South American country or something. And I've heard this from everyone with every language is like you can take every advanced language course in college, you can go into the fine nuance of this and that, and the upper level and the upper level. The best way to go is just go live in another country and just everything about facial expressions to different, just different vibes, different norms, all lead to a better. Now I'm an idiot and I failed German 3 straight semesters in college, and I never did that. But I'm taking it from what I've heard other people with different language. They all say whether it's Spanish or German or Chinese or whatever. That's kind of what you're saying is like, so we've got this, it looks good, but you go in there and then there's electrical signals, and what about like the hormones, and what about just someone getting drunk? Is there ethanol in there? And it's like, but like a language, you don't have unlimited funding to send every student to South America. So it's like, hey, like it's like 95% accurate that like these are the verbs, these are the adjectives. Like, sure, maybe there's some nuance like at a bar or something, but for the most part. And so you have to find something that's worthwhile pursuing and then kind of double and triple down on that. Correct?
Yeah. Yeah. I mean, I think it's a really cool technique. That's why I like tissue engineering so much, because you can isolate some of those, right? If you have a hypothesis, if you're wondering if it's going to be the white matter tracks, or you're wondering, is it the biomaterial, the material that's coming into contact with, well, you can isolate those different variables. And that's what I liked. And then you can find out, if you want to, on molecular level, how those cells are responding to those changes. So you can run all kinds of tests. And what I like to say is, right, you have to, if you run one test, you have to validate with another, and it keeps going, right? If you're, making a really good sound study. So, if you wanted to see, if we're looking at molecular, what's happening molecularly, right? You could do imaging, right, as one, and then you would validate with doing some more expression techniques like Western blots, all of that. Like you really, really, want to make sure, because what you might capture with one technique might not relate to the other, right? The other thing to think about is when you are taking cells out of that environment now to run an assay, they're already starting to change.
Yeah. You said
So,
you said you guys did that with just to like already you would see changes, right? Because you'd heat it up for cross-linking and then you'd submerge it with fluid so it wouldn't, I think you said, wrinkle. So it's like, the second you do something, it's changing, right?
Yes. Yeah. Yep.
I mean,
so there's so many factors. And we're never, so if I were to create if I were to test the cell in that outside environment, and I'm saying, oh, well, this is the way it works, and develop a therapeutic off of that test, that wouldn't necessarily be accurate, right?
Yeah. Bad things
because
happen when you rush shit. Yeah.
So yes, we know that.
Yes. But so all right, well, kind of like unavoidable black hole then. Everything we're talking about, even in your paper, like towards the middle, I realized just so much of it was just process, right? It's like 10 millimeter, 10 millimolar, you got to do this. We're using that. And even when you're talking about, that's what I noticed is like just the images. You were like, go to file, you open up. I was like, oh, you're truly going in and doing everything, right? Which is what you do when you have an airtight argument. And I'll timestamp it in case you want to splice all of this out. So it's right at 8:30. It's right at 8:30. So we can cut the rest of this out. And now as we go into Zivilboten,
no, we're having a free discussion.
We're having a free discussion as people who are not associated with any universities. We're associated with Tommy University. You clearly show, like, you're not the first person to do a PhD thesis. You're the first person whose theses I've read, but I would imagine that they're all probably that in depth. You go in and you show everything. Why? Because you have to show airtight. This is what I did, as Dr. Bhargava said last week. You don't just say, well, I used a 10% solute, because someone's going to say, why not 1%? You have to go, well, this is why I did this. And you kind of have to block them off the past to show that, hey, it's airtight. You can question me on anything. You can break it all apart. Don't worry. I don't care. TSA, put my bag through the x-ray scanner. I don't give a shit. There's no bombs in there. Like, go for it. So so that being said, so you're not the first person to do a thesis. This is clearly what has become the norm and what's been expected. Why? Because it has to be able to put up with every critique and evaluation.
Yes.
Now, how insane is that when you forget Martha Fowler, when you have individuals raising the same questions? It'd be one thing if it was just you and all the doctors were like, dude, here's this just young chick. It's like, it's like when you're 16 and you're all angsty, you're like, there should be world peace. And your parents are like, dude, you don't know what it's like, get a job. And you get older and you're like, oh, sometimes war happens because, okay, I get it. There's resource scarcity. We can't all hold hands. Like, but when you're 18, you're like, dude, we're just killing each other. It's not even like that, though, because you do have the McCulloughs, the Malones, the Bargavas, the Farids, the Nass, the Ursos, the Merricks, all coming out and saying, and not just research doctors, correct? Dr. Malone with DARPA and Defense Threat Reduction Agency, Dr. Bargava with UCSF, Dr. Malone, the most published cardiorenal physician in world history. And they're saying that at the very least, you have to question it. And if you notice, none of them are definitively saying that like these shots are bad. What they're saying is we don't know. That's all they're saying. They're saying we don't know. They might be wrong. People might look back at the episodes I do with them and be like, these guys have no idea how wrong they were. But all they're saying is these things need to be questioned. So where does that sit with your mind, which led to you reaching out to Dr. Bargava and coming on here? Where does that sit with your mind when everything you just showed that in that 78 paper or the 78 page thesis about how you got to argue everything, everyone's like, yeah, airtight, back it up, sources, footnotes, everything, nice skills and schematics and showing the diagrams and the artificial coloring and here's how I built my little at-home thing. You've got the plexiglass and the little motor panel. If the ATF came to your house, you might go to jail. They think you're building a bomb. You go in and show everything. But right, you have to go in and show everything.
Yeah. Well, that's important, right? I mean, to even say that I was sorry.
Well, I was going to say that I think we have a delay. To say that when you start questioning anything else and all of a sudden everyone around you tightens up or the fangs come out and they say, shut up. And you're going, guys, I'm not even making my own thesis. I'm just saying, what if X? What if Y? Even you're saying with this, you're like, we're still, even you were hesitant to say the word validate. You're like, we got to check. So when you question something else, like what's going on right now with COVID, with shots, and again, we can slice all this out because I don't want you to lose your job. How does that has to rub you the wrong way, right?
Yeah. I mean, like I said in the last episode, as well as, saying it now, when I did this thesis, and it's not perfect, you saw the picture, we're being transparent about the machine we're using, you see that, like you said, the plexiglass, well, because the thing is, you want people to be able to replicate, right? When you can read the other articles that I've published too, right? You want to be transparent about your methods, and that does depend sometimes on the journal you're publishing in, but you still have to show, right, so some transparency of that, what you're doing, and you're displaying what you did, and someone might look at that and think, oh, well, why didn't
I can pull up your, I can literally, transparency, I can literally pull up your studies, beta cycle. Sorry, keep going.
Yeah, well, because you want to encourage growth, right, in the field. So what I'm saying for the tissue engineering stuff, if somebody reads my thesis and looks at it like, oh, they could have done it this way, why did she spend so much time, or they look at our model that we built in the lab and they think, oh, well, they could have just easily done this instead, and these people will probably get a better result, or they look at the different models that I developed by going to Home Depot and building all kinds of things. But I want to be transparent about that as well. It's something that, we make in the lab, and you want other people to be able to replicate it, because that is also going to help show that what you did is contributing to the field, right? That you're allowing the field to move forward. So, to to me, what's going on was so strange, because going through a thesis process, even going through articles and understanding data and how to present data and how you have to keep your data, and it's something that is sacred, and you display it to the best that you can. You're never you don't want to be hiding something and say that, oh well, like if, for example, in that thesis, oh well, I solved the problem, I think if we just target the axons and we'll be good. It's like, no, that's not the case. You can't just pick something and decide that's the way it's going to be. Especially with biomaterials, or something that we don't understand yet. So even in nanomedicine, it's something that is, or has not been through the trial phases, as we know, for a vaccine, right? This is a new development, and that's why it is so rare, and that's why it deserves questioning. Especially, I mean, everything deserves questioning, of course, in science, but when you're developing something that's brand new, even in the labs I've been a part of, right, it takes time and lots of trials and lots of error, right? It's never usually right the first time. That's it's not really possible.
No.
Yeah, so there's I was having a conversation with someone, and they brought up the point of, well, if you understand data and you go through data just with practice, and you look at the studies, everybody's, oh, look at the trust the science, all of this. Well, if you bring the scientific process, right,
there's no trust. There's no trust.
It's
verify.
It just made it very difficult, right? So we know that the polio vaccine took a long time to get where it is. It wasn't good on the first try. The first trials, nothing is ever good on the first trial. So we know now that we have better technology, but that doesn't mean we can do this, right? We can't snap our fingers. I can't just create, now that we have 3D printers, I can't just create an organ, right? I can't just make something that I want to. It takes a process. You have to test each individual component of what you're making and understand the influence of that on the host or on whatever system you are experimenting with or investigating, right? So we know that in my paper, I was using these different biomaterials, and in my other papers, you'll see the same theme where we talk about the biomaterials as a factor. And so you have to be transparent about each one of those, because each one of those could then have a different reaction in the host. So what the result you see might not be necessarily due to your therapeutic. It could be due to a piece
materials
of something. Yeah, it could be the biomaterial. It could be a combination, as we said before, of everything. I mean, now, right, the other thing is that they're using a variant from, right, the Wuhan variant, right, to make these vaccines, right? And then after the vaccine development, there was Delta, now there's Omicron, but we're still going to give reinfect people with the first one. So that's why they're getting all the others, right? Does that make sense? Because that's a new variant, right? It's similar to the flu, whereas we can say, right, if you get the flu shot, there's a chance you still might get the flu, right? Because of all, because there's different variants, it's going through different people, right? So we have the Delta and we have Omicron. So you're getting 3 different injections of this one, right? And you're getting these bad responses, right? Which, what I kind of think is that you're putting this very high dose because you think that this particle is going to release slowly over time. So you think that the person's body is going to acclimate, right, to this process. Where if you were to just inject that without the nanoparticle, you probably get really sick and the reaction would be immediate. Wouldn't be good for the host. So that's why the second time, right, that they inject these particles, that's why I brought up the antibodies that they've shown, right, the anti-PEG antibodies that can attack these particles and degrade them, right? And now you have a we call it a burst release, sometimes when that happens. So you have a burst release of the of the mRNA, right? And the other issue is that we are still trying to understand how these biomaterials distribute and clear in the body, right? So we don't have all the answers. It's science. That's why they call it research, not just search, right? You go back in
again and again and again.
And again and again. Yeah. And you
Again,
isolate the
again.
variables, right? So, I mean, we know that there were only, 3 primates in their trial. And yeah, there's, you have biological replicates, you have technical replicates. I mean, there's so much here. And, when you look at a thesis, and I appreciate that you did, you can see that you have to say what your sample size was. I did this replicate this many times. Yeah, you have to, and that's
everything.
Even in research articles should all say that. It's something that in the scientific community we can't let go of, and that's why I got so frustrated with this, because to me it was almost like a cop out, right? We work this hard and we generate data constantly. We're working in the lab constantly to tell people now. Or on the Candace Owens show, she says, "Oh, imagine now all these high schoolers. Oh, you don't need to develop a hypothesis for your project. You can just do it.
This is what it is. It's
what it is, because I said so, right? Because
yes.
I said so. So if I say so, and it is what it is, and that to me was, when people were asking me like, why are you getting involved in this? I said, well, because I feel like science is going down a dangerous path.
Yes.
As someone in training, I'm not going to say, oh, my stuff is the best. It's, it's the one thing that works. Oh no, somebody could have developed a much better model to look at tumor movement and migration of the brain. Mine is not the best. Like there's always room for improvement, and somebody could have done it better. That's fine. That's we're all learning, and that's what I need to be transparent about, because somebody could be doing something better, not even for brain tumors, maybe for something else. And then we realize that, oh, now you can apply this to tumors, or maybe this model will work better for this case. So yeah, I can't believe where we've ended up. And the fact that it has to take courage for someone to talk about it. That's like somebody, this may sound bad, but it's like somebody be happy for me for talking out about the work I did in a paper, and being transparent about it, or questioning a different article that
She's
somebody
so
else
brave.
did.
She wrote her thesis, and it's like, no, that's not only is it not brave, not only is it not normal, it's expected. As a matter of fact,
It's
it's the requirement.
That's a better term. Not brave. Sorry.
No,
yeah, brave would have, but you're going against the
no.
whatever's been normal.
No, I think it does.
Like,
I think it's the right
like the expectations, like you said, expectations. You're changing the expectations, right? So if you would just let a PhD student go without them defending their work, right? So we have a defense that we have to give, a public defense and a private defense.
You literally defend your thesis.
We literally have to defend our
get the sword out. You have to defend the thesis.
Yeah, no, you have to defend yourself, with science.
Yeah.
and you could be wrong. I mean, that happens. There could, there's always negative data. It's never going to be 100% good. You still present everything that you did. But we have to, bring ourselves back to that with these kind of situations, right? Because the moment we start, because when you think about all these people that are getting trained right now, like myself, that are now learning that you can essentially be a part of a pharma company and develop designs and won't be challenged.
It's raising the
You're
kid saying
changing
one thing
science.
and doing
Yeah,
another.
you're messing with, you know, the backbone of science, right? It's like I said, it's research. You know, got to go back and look at it. We know that we know things from this trial. Why can't we accept that and move forward, right? I mean, if, from these trials and what's been going on with these vaccines, it would be like a PhD student that presents their work, or anybody that presents their work, and there's something off about it and they get questioned and they just shoo it off. Oh, no, it's
Trust the science. Shut up, doctor.
yeah,
Trust the science.
yeah, yeah, exactly.
Well, to me, I think kind of like the pertinent core to all of that is what you said, is it's science, right? It's the same thing when people ask me, Tommy, why do you do? I don't have an interest in COVID. Yeah, I was a biology major. Yeah, that's not, but this isn't my thing. As I said to you beforehand, I'm like, I'd much rather we just start talking about JFK and like an alien. It's like, that's much more fun. But to me, it's something much deeper. And what it is the ability to speak. Am I going to talk about your thesis today? The guy I had on before you, we talked about the Indian concept of the yugas, the cycles of human spirituality. And the woman before that, we talked about the armaments building up on the borders of Ukraine. What it is speech and the ability to speak. I love, I hate. I am for Trump. I am for Biden. I hate Trump. It's the ability to exercise all of it. Do I want to do nothing? Do I just want to sit here and tell a story? What it is speech. So whether it's COVID or whether it's anything else, the defense budget, civilians killed in a drone strike, the accessibility of clean water in Congo, what it is speech. That's just what it is. So that is why I am up in arms about it. So when someone says, Martha, why are you getting in here? What, this isn't, stay in your lane. No, you're literally defending your thesis over 78 pages. Yet you defend the, is it about brain cancer? Is it about the aerodynamics of a new reentry capsule? Is it about a new type of earthquake resistant cement? It's defending the science. So although it's in something across all science relatively close to you, right? Biomedical, it's still what it is it's the process of science. So someone might say, oh, well, it's just, well, no, yeah, sure, it's close, but it's within really close to what you do. And it's the process of science. And sure, there's a little bit of like hey, I had to do this, but you have to do this too. And that's kind of like the ego talking. But then there's like, but this is also what has like yielded this. That's what has yielded this society. Yeah. Is the ability to have microphones and IMAX and clean water and gigabit Ethernet, is because of that over the centuries, just the defending of the thesis. Just you got the sword out and you're like, I think this is what's right. And that is what moves us forward. That's why we have the antiseptic technique. The butchering art by Lindsay Fitzharris talks all about this. Joseph Lister saying, hey guys, I think we should clean off the scalpels before we take it from one wound and put it in another. So to me, it only makes sense that you'd be up in arms about this, because you know what it yields. You're clearly passionate about this. And as you said, that humility. Yeah, maybe someone else has found out a better way to discuss like tumor migration. Good. Let's get to the answer. Sure, it'd be great for it to say Martha Fowler on it. Yeah, of course. But it's also like, let's just get to the answer. We're all going to benefit. So when it comes out that it's settled, you guys can't talk, it's settled anymore. If you had said anywhere in your paper that the science is settled and this is the answer, I probably just would have not done this podcast. I would have been like, hey, dude, I think you got some personal growth to do. No offense. Right. But you didn't. The entire thing is, and these are the limitations. And these are here's what I built. This is how I built it. It would be better if we did this. it you have, and to me that's what clarifies why you see people like Dr. McCall, Dr. Malone, Dr. Bhargava standing up and going, hey, what the hell is going on? So I'm sure the last 20 minutes has probably now cost you your job, so we can cut that off if you'd like. But yeah.
No, it's all free speech. I've had this conversation, and I am more than willing to have this conversation with anyone I work with anyone who's around me. I'm serious.
I know. I know.
Yeah.
Yeah.
Yeah, because it should happen. Yeah, it's not, oh, you now talked on a podcast. Well, I didn't say, I'm just talking. We're talking about science. And it's been interesting. The people that I have, I've had a couple people, say, this was before the podcast, early on, when I was investigating it and wondering what was going on. I'd mentioned it to some colleagues, and they did, oh, what, you're an anti-vaxxer?
Oh, you anti-vaxxer, Martha.
Yeah,
I always knew.
That's the immediate response. And, I said, no, of course, there's, the polio vaccine. No, of course I'm not anti-vax. We know that one has taken a long time. And someone told me this one thing, well, if you got the polio vaccine and then you got polio, would you still be game for that vaccine? Right. So, yeah, I'd have these conversations with them of, well, we know the story of polio and how that took a while. And, we know how to get there. And the way to get there is not, this is what it is, right? We knew that even back in the day, they noticed what was wrong, and then you try and change it, you make adjustments. And then of course, mistakes happen in science. Very unfortunate, right? Like in the polio vaccine, right? For instance, in the beginning. And so, but when I was able to sit down with these colleagues, and explain why this is why I wasn't so for it, or why I was at least just questioning it. And I never influenced anybody or told anyone not to, right? Because that's your decision at the end of the day, and it should be our decision as well, right? Each way, if you want to, if you don't, who cares? You go get it. If you want to, take ibuprofen.
If you want to pray to the sun god, it doesn't matter.
Just
do whatever.
Go do your thing.
Go do a rain dance. It doesn't matter.
Yeah. It's awesome. Yeah.
But just don't make me do the rain dance.
But and I think the thing too was the quick judgment. When you sit down and you talk it out, you can, if someone's willing to listen, of course, too. But the people that I was able to discuss and have an open discussion, they did, then understand where I was coming from and why is more concerns when, I talked to them of, this is what we do. This is like, come on, this is
literally what, it's one thing if I sit here and go, guys, this is how we get at the truth. You're literally getting your PhD. Like, you guys are in the business of ruthless examination.
Yeah. And I still have a lot to learn too. I'm in training, but that was the hard thing, was being in training and having people.
But to me, I think there's the most beautiful symbolism though. It's like everyone else we're just doing the thing. We're just taking the class of PhD. You did it. Have you ever seen Willy Wonka or Charlie and the Chocolate Factory?
Yeah.
Do you remember that?
Yeah.
Do you remember when, who's the guy, Slugworth, and he tries to tempt them all with and at the end Charlie's the only one that didn't it, and he's like, you were the one.
Yeah.
You are practicing what you preach, even when you don't know it's a test. So you can go through and what he's don't touch anything, whatever, don't talk to the Oompa Loompas or whatever. But he has actually shown that he put his money where his mouth was. He didn't know it was a test. He was just like, hey, dude, I can't give you Mr. Wonka's gobstopper. This is just, I'm sorry. To me, there's such a poetic beauty in that, to where everyone is, we're all questioning the science and getting our PhDs. And then you were like, yeah, what about that science? and everyone is attacking you, and you're like, no,
Mm.
to me, it's like you're holding on to the gobstopper. I think that's what's so great about it, is like you, in a sense, like you kind of earned it with, like, forget the actual PhD. Like you just showed, like, I don't care what everyone's saying. I don't care what it's on every news media. I don't care if there's a little blue label under every video that says the word COVID. What am I seeing with my eyes and my ears and my own functioning brain? And I don't care about
Mhm.
the group think and the peer pressure. Guys, this isn't right. And to me, that's bad. Now, granted, you're probably gonna lose your job now because you've been on this podcast twice. But that being said, you have my
Um,
applause. Yeah.
that should not be the case.
Yeah.
But well, and the thing is too, I mean, I don't know how much I should get into this, but I did have people that I was looking up to um tell me that this was because I thought this way, essentially my future was doomed, right? So you're hearing these from people
Wow.
that that you had looked up
Ugh,
to, right? So
that's
it's yeah. So when somebody who's in, and that's the whole thing, right? You have people in these positions. We're seeing that now. You have these people in positions of authority, and when you're hearing these these things of just, well, we should be able to talk about it. And there were points where I would listen to the other side and I would understand, right? So there was this back and forth. It wasn't I was um trying to attack anyone or anything like that. It's good to hear the other side as well, and it's good to look at those articles, look at what Moderna did, right, and critically assess what was done. And so, if we were going over these articles, as I remember with spike protein, talking about that, and asking questions about the antibody titers and just trying to understand it, wrap my head around it. And that was all fine. But then when it came down to the moment of, hey, this is getting more serious,
sinister.
that's when people started to it's interesting. You even have the colleagues, and I'm not trying to make it sound like I can handle it.
Yeah, no. I mean,
It
no, no.
wasn't, yeah, it wasn't, because this is what we should do, right? So in science, you're gonna have
Take the
people, like I
flack.
said, during lab meetings, who are who will be like, oh, why did you do that assay? That's so dumb. Like, why would you do that? You should have just done this. They don't
Yeah,
use so dumb. I threw that in there, but
yeah.
I
You
don't use those words.
just like they never
But
said that. They're like, you're so dumb, and I hate you, and you're laugh, and
yeah,
you're like, dude, what? But yeah, no,
yeah.
you have to be able to take all those things in stride. No, I get you. I
in,
told you
in
before.
science, right? Yeah, I mean, because that's people are always going to be critiquing your work, and you pour your heart and soul into the work, right? And
Of course,
because
you're going to be a
and
little
hours,
associated with it. Yeah, it's yeah,
yeah, yeah, of course. But you have to be able to take the heat, right? Take the heat or get out of the kitchen,
yeah, yeah. Mic
So, and
drop.
that goes, yeah, and that goes for everybody. And it was because before, in my previous lab, or a lab a while back, I did have the opportunity of seeing some of the industry side too. So it's not like industry is bad.
Of course
It's
not.
not, there are these companies that are doing great things. I've been able to see that. There's, these groups that, I would want to be a part of, that I was, and then just somebody to, to say you're not gonna be there now because you're questioning a trial.
It.
Yeah,
I
that's,
mean, yeah. The most
I
I
don't believe
Yeah, no, I don't. No. You will be proven right in the long run, and it will pay off dividend. Yeah, no, I am the most like wildly pro capitalism. Like I'm like, go print money, go ruin the competition, like crush the world, like take what's yours. Like I'm all about that. I think that's what moves humanity forward. So it'd be one thing if I had like the communist flag behind me and I was like, these big pharma companies. No,
it.
I'm all for it.
Yeah,
It's
yeah.
but
Because there's, but I mean, it's still right. Everybody needs to be held accountable and kept in check, right? I mean, you would want these when you think about, company, I've, met some of these people, seen some of these companies, and they're great. I'm not talking about these big ones
Sure.
that, with the vaccine. I'm just talking about some startups and things like that. And it's been great to be a part of, and you see the big difference that they're making. They're creating these awesome devices and everything. And they are challenged of, people are questioning them. They have to, go through a lot of regulatory processes. So now doing something like this, right? That's one of with the vaccine, I don't think you realize that they realize the ripple effect, right?
Yeah.
On how this is going to change so much, because you could, right, if all things don't go well, you're also now causing distrust, people's distrust now in companies and in science and in research, right?
Well, how come I'm it's the analogy they use. Sorry to cut you off. It's the analogy like, and I think Duncan Trussell, the comedian, used. He's like, you grow up and your parents and your teachers are like, don't smoke pot, you're gonna overdose. And then you finally do smoke pot and you're like, that wasn't that bad. Well, now the same people that are saying don't shoot heroin, you're like, I bet they were lying about no, they actually weren't lying about the heroin. Like, so it's the same thing. Well, why should I trust the FDA or the CDC on this? Why should I trust anyone? But then furthermore, it's the distrust in everything of everything out there. Well, how do I know aspirin's perfectly safe? Tommy, aspirin actually is perfectly safe. Why should I trust my doctor? They say they're calling it a clot and I need to go get a scan. And it's like, no, the doctors are telling the truth. There is that ripple effect in everything. And then not only that, it's also the sort of, well, he got to do that. How come I don't get to do that? Well, and we're creating this now entitlement where it's like, okay, brand new technology, mRNA. What else is brand new? Self-driving cars. Well, hey, trust the science, guys. But that's exactly the point. Is it ripples? Sorry, I know I interrupted you. That was rude of me.
No,
but it's the ripple effect. It's all gonna come around. It's gonna come around to everybody, whether it's losing trust or whether it's other companies being like, hey guys, regulations don't matter. Let's roll. Like, we're opening Pandora's box.
Yep.
I know, just kind of rambling there, but
no, that was really, that's because it's, it's true, and it's something to be concerned about. And I think that's why it's awesome that others, as we've been saying, are speaking out. You have these people who were involved in the mRNA technology. You have, McCullough, who I remember he was one of the first people I saw at the Senate
Texas
hearing,
Senate,
and in Texas, who why not treat, why not treat, and and everybody just automatically. So when you experience that, right, everybody, I think that's why people are afraid too, because the reaction is so inflammatory. It's
vicious, reflexive.
People just, and there's really not a need for it, because it's okay to critically assess, right, anything. Even when you watch a documentary or anything like that, you should, critically think and analyze what is happening and process. And it's okay if you're on one side or the other. I think as we're going, we're talking about this, you still just should have the conversation. It should be okay for you to talk about it. It shouldn't be, such a scarlet letter. For
it's
just bringing it up.
It is. There's this sort of punishment by example. When one person stands up, you say, shut up. Because there are a lot of people with very real, hey, I got to provide for my wife and kids. And you know what? I don't like what's going on with COVID, but I'm going to shut my mouth because I'm a orthopedic whatever. I mean, I get that. That's a very real thing. But the beautiful part is that it's the St. Augustine quote. Truth is like a lion. You only need to let it, you need not defend it. Only let it out of its cage. You only need one McCullough. But we've got a McCullough, a Malone, a Bargava, a Fowler. We've got a ton of people talking out. We've got a Rogan, a Fareed, a Nass, that it will come out. And then the attacks matter less and less. If one person goes, guys, I think gravity is 9.8. Oh, shut up, Tommy. All it takes is enough people to go, what, they're like, what is that? How come it did go down? It's misinformation. You don't need to defend it. Like you said about the cells breaking open and like the necrotic centers coming out. It is an acid. It's going to burn through. That's the beauty of truth and science. And it's not even like a political scandal or a secret that can, burn all the files. It's gone.
Yeah.
It's just science. It's the physical reality we exist within. It's just what it is. It's just what it is. And no, I think you have to speak up. And I think it's shameful that other people don't. I mean, I get it. I do. It would be stupid for me to, it would be detached for me to say, how come you're not speaking up? As a 31-year-old with no kids and no loans? Like, it's very easy for me to, I make my own schedule. It's very easy for me to say what we should all do. I get it. There are people with jobs.
Yeah. Oh, it's
real. I get
scary.
it. It's real.
Yeah.
It's terrifying. But if we don't do it now, when?
Yeah. Yeah. That's why, I appreciate that, you've had these different scientists on, because, even just listening to them of, this is why I am taking a stand, because this is what I've been trained to do. This is my training. This is not abnormal to be discussing this and, critiquing it. And as I always say to people, well, there are journals out there. You can look at, it's not misinformation. It's been done by other scientists, evaluating vitamin D deficiency. That is out there. The antibody pig, the anti-pig antibodies that I brought up, this is also being evaluated by scientists all over. So, and the same thing, there's now articles looking at the antibodies responses actually to the different variants and how those are different, how the reaction, the neutralizing antibodies for, a friend sent me this article and it was quite good, how the antibodies that your body will create for that first vaccine that you get are not going to react the same to other variants. And that's why you'll still get these variants. I mean, this is what we're saying here is not information or misinformation, because it is out there. The research is being done. And that's the thing too, right? There's going to be more research and more. And that's perfectly okay. People should also now be allowed to study the effects of mRNA and how this could be causing bad things or good things, right? I mean, there needs to be more research on it. And the more we see, the more we're going to know and feel more confident about whichever way you lean on. And that's fine, and it's interesting that we'll listen to, maybe I'm just rambling now, but
no, keep going.
We'll listen to one scientist's view,
Dr. Fauci,
and not the other, right? So just for an example, like when you're writing a thesis, you look at, or just a paper in general, you always have to, I actually, at the end of articles, you make that disclaimer of these are, this is what could be wrong with it, right? These are the limitations, right, of the study, right? So you have to say that, and you have to understand that it's not the best. And when you do your research, right, if I'm looking at one opinion versus the other, right, I take into consideration both of those when I'm developing a study, right? So if one thing is telling me that applying, I don't know if this is too good an example, but apply this material versus this material, right? You look at the pros and cons, right? If you always just choose, quickly choose one, you're hearing one thing, okay, well, I'm just gonna stick with that. There's always something else, right? You have to do your research. You should see both sides. And that's, I'm open to hearing the other side. I always say, if you want to have a polite conversation about it, I'm okay with if you just want to talk about it, but I know that some people get really emotional about it. And I understand, one person had said the card of, oh, well, people are losing people to this. How can you not be more sensitive? And to me, yes, that is unfortunate. We lose people, I've lost someone to cancer, for an example, but that doesn't mean I'm not going to critique cancer therapeutics,
Yeah.
right? I want better for those people, right?
Yeah.
So I'm going to try and push for better. I want these people to recover rather than relying on something, right? So say there was, especially cancer treatments, there's for certain cancers that are very aggressive, lifespan of after diagnosis, 3 to 5 years for some of these more, serious and aggressive cancers. And there's not hope for these people. That's, we need to develop and need to work on those. And that's why I mean that science is important. We need to continue working on it. Where then the opposite side of, oh, well, I've lost this person to this, and I understand that's unfortunate. Let's want better for these people, not something that's a vaccine from the very beginning, right? Where now these people are continuously getting sick because you're injecting them with that one and they're having a reaction over and over, while also having reactions to other variants, right? Getting other variants. So constantly being sick. We should want better. We should strive for that, and that should be okay.
I talk about all, I think I told you, and I've talked about it before on this podcast. My main goal of this is to make a ton of money so I can put it towards mental health research, because as MAPS has shown, with the application of LSD or psilocybin or mescaline or ibogaine, you can wildly improve overcoming PTSD, or just, I guess, almost organic depression, if you will. I lost a brother to suicide. I have his initials on my shoulder. I wear his necklace every podcast. Me critiquing it is not saying, Tommy, don't you know there are people out there? I get it. I do get it. That's the very reason why I'm pushing. And so maybe you haven't lost someone to suicide. Maybe you haven't lost someone to cancer. So then what? Do you still have a right to defend that viewpoint? Well, above that is the literal, and now it's like the flag billowing, but it's the literal application of freedom of speech. If I see someone make a joke about suicide, I don't necessarily like it. I just don't look at it. So if someone says people are being lost to this, and I said this, and I think this is really the best way I could summarize it. To everyone saying that Joe Rogan, or technically 6 months prior, Tommy, if anyone's saying that Joe Rogan's spreading misinformation for interviewing Dr. Malone and Dr. McCullough, my question to them is this: were you tricked by them? I don't mean like a kid. I mean, were you tricked by them? Did they dupe you? Did you take some horse pace and you ended up in the ER? Did you not take the vaccine and now you're dying from COVID? Everyone, to a T, will tell you no. If you ask why, it's because they say, I'm not an idiot. But you know what? The misinformation is hurting. So what they're passively admitting is, I'm good. I know Santa's not real. I know there are no monsters under the bed, but you're spreading misinformation for all the other idiots out there. So what are you implying? The government needs to step in, or someone needs to step in and stop that, because the masses are too dumb to think for themselves. So what you are now passively implying is they don't know what's best. They can't look at it and decide for themselves that it's not real. You don't need to censor Scientology. If someone comes and pitches Scientology to me, I'm gonna be like, hey, yeah, interesting. You're crazy. Like, right? But you don't need to censor it, because I'm an adult. I can go on YouTube and I can
You
watch
can make your own decisions.
I can watch a documentary about how Queen Elizabeth and aliens did 9/11. Fun, sure, but I'm not losing my mind over it. So if you say, I'm not dumb, I didn't get tricked by McCullough or Malone or Fowler, but everyone else is, what you're saying is they're too stupid to think for themselves. I need to decide what information they have access to. And that is far more lethal than any information could ever hope to be. And that's where, so don't you know people have been lost to this? Yeah. I'm sorry.
Yeah.
It's what it is. It's
Yeah.
what people are also lost to putting leeches on their body.
Mm-hmm.
I'm sorry.
Yeah.
Like, it doesn't mean I don't have a heart, but you can't science is brutal in that manner. You can't hide behind an emotion and go, don't you know Bob lost his grandma to COVID? I'm sorry, but you
Mm-hmm.
can't stand behind that. That's not, feelings don't matter when you're bringing up how do we develop something that can survive re-entry from orbit. It's, now I'm rambling, but yeah, it's, that's what it comes to though, is what you're saying and what I'm saying is we're not even making hypotheses. We're saying we could very well be, we might look back in this in 10 years and go, oh my God, I can't believe I was against it. You remember when everyone
Yeah.
took the COVID vaccine and then life expectancy went up to 250?
Right.
Oh God. I'm open to that. I'm open to
Right.
that.
Right.
What I'm
We're
saying is we have
you're
to
an adult. We're
discuss
making,
it.
yeah. Yeah.
As you
That's
can tell, I start getting red in the face, start screaming, and
no, but it's very true. And I think, again, having these discussions, just expanding our knowledge and our minds and challenging ourselves, that's for me with this work is also a constant challenge, which I like. Like I said, I'm still learning, right? Every, it's
everyone is.
everyone is still learning. And it was an interesting point that I was actually discussing the other day was how now I believe it's in California they're making the age now 12 years old to
Consent.
consent to a vaccine or medical decisions, right? So, but the issue with this, right, is okay, so you're giving children this freedom of choice, but you're not giving adults this choice.
That's a good point. That's a really good point, actually.
Right?
I'm gonna
And
steal
to me
that.
that is it's so backwards. Because we've gone through where we should have gone through, you learn how to write, how to read, how to write papers, how to form arguments, how to research things, right? With age and all of this takes time, your development, everything takes time. And so, when we get to this age and we're being told, you have to do this, whether you like it or not, but these kids, they can
They
do
can do
whatever
it.
they want.
They can do it. Yeah,
Yeah,
Tommy,
they can do it.
You're not, listen to Fauci, Tommy. You gotta get you gotta get the vaccine. I'm sorry, I know you're 31, you're an adult. You can't do this. Billy over here, though, that kid's got a head
Yeah.
on his that kid's got a head on his shoulders. Think you're a girl? Screw it. We'll chop it off. You want the vet? Come and get it. Come on in. Come on in. It's harmful misinformation, but yeah, I mean, but again, the beauty is it's not a political scandal or a crime, a financial crime, something that is it's made up in the human world, and that you can burn all the files and it doesn't exist anymore. This is just science. It will
Mhm.
it will get out. The acid will eat through the wall of censorship.
Yeah.
All we can do is facilitate it. It's science doesn't give a shit. It doesn't care. It is the lion. You just got to kind of open the latch and just be like, and here we go. See what happens.
Mm-hmm. Mm-hmm. Yeah. Yeah.
Closing thoughts, Miss Martha Fowler.
Yeah.
We just burned through
Oh
2 hours.
boy, I know. Oops. Sorry.
I don't care. I don't care, dude.
I figured we would probably just be able to ramble on. But it's all
We can keep.
it's all good information. And no, I feel like it's
we can always cut out the last 50 minutes if you'd like to keep your job.
I don't know if we should. I don't know if people are gonna be sick and tired of Tommy and Martha won't shut up.
Dude, trust me. Those who are gonna watch are gonna watch, and those who aren't are not.
Yeah, this is true.
It's just what it is. We can. I mean, listen. If you're terrified, if you want to wait till you get your PhD and then release the extra hour, it can be like the bonus session.
Yeah, right.
That's
Yeah.
that's
I
genuinely up
mean,
to you. I'm not even kidding. Like it has zero
mhm.
effect on my life.
Well,
It actually does
I
affect yours.
I appreciate that. I do. That's that's good. But mm, I don't know. I think yeah, maybe I will. No, I'm just kidding. I to me, if I would lose any, we've said before how you lose friendships, you lose people for having this discussion, and that shouldn't be the case. We should be able to openly talk about this as we talk about our likes and dislikes with other situations and our own medical choice and decision. And I mean, with all the data coming out as well, people are seeing, are seeing this data. And the the population I do worry about, we said for children, and also young healthy individuals, right? What is this repeated uh you know vaccine, as well as getting sick with everything else, right? Everybody's going to have Omicron. What is that doing to the body? Well, that is going to take, right, 10 or more years, long-term studies to see. If just speaking of, thinking
Yeah,
scientifically, we're going to have to wait to see those effects.
yeah,
To say that we know the long-term effects, that's completely false, right? We don't, there's no way, as you said, the
can't.
example of the pregnancy, right?
Yeah,
There's no way to know. And, for people that, I've seen Dr. Bhargava, which I thought was very inspiring, talking to the people that had the adverse
on
reactions or
on
adverse.
Aubrey. Yeah,
Yeah, because those people were previously healthy, right? So that's why this should be someone's choice. Someone should not be pressured into it, because you think about people who are even, let's say, policemen, firefighters, EMTs, right? Where this thing could affect their heart, right? These people that do need to, be active or are put in serious
Navy
situations or
Navy
dangerous.
SEALs.
Yes, yeah. Oh yeah, military. Yes, of course.
Yeah,
Yeah,
yeah.
of course. And so these are young, healthy individuals right now, where we know they'll be okay. Omicron's going around. People, these young individuals will get through it. Most likely. And so being able, or forcing something that we don't know anything about on these people, is that to me was just a red flag. And kind of because you can imagine these people, when we see these adverse reactions or adverse events, they thought that they were trying to do their part. Everybody was
Yeah,
saying, do your part,
yeah. Do
get
your
this,
part.
you have,
Hey guys, I had the
yeah,
side effect.
yeah, everybody do your part. Well, it's like,
Except
well,
for
now
you.
I can't. Yeah. Well, now I can't, have a living because I have this situation, right? So it's it was complete, when you think about some of this, it's complete opposite of, oh well, you have to have the vaccine in order to have your job and get paid. But if you now have the side effect, you can't do your job, well, there's no liability. We know that's a fact, right? There's no liability for these companies. They're not going to pay for your support, which is not
And they're
right.
shattering
Also,
profit records.
Yeah, what, 36 billion was it?
36.5 billion according to Axios as of
36.5.
yesterday.
Yeah,
No incentives though,
yes.
guys.
And
No,
now
no
the
incentives.
and now the data is coming out too, or at least now we have some people on network saying, oh well, masks should be an individual choice. And all these things that were, as we've said before, were misinformation. Side effects were misinformation, but you don't know something's misinformation until you see it long term.
Yes.
You can't right away.
Yeah.
I mean,
That's the
people
whole
are
point.
coming up with, right. So if you're seeing, okay, all these, these people are having side effects, oh, that's misinformation. Well, this is just the beginning population, right? This is the beginning of the vaccine. These are people that went into the trials. This isn't after everybody, starting to get it, right? So we might see things down the road even more. We don't know, or we could not.
Yeah.
But only time's going to tell, right? I mean,
It's socks too, Oleg too. That's misinformation until, well, no, now it's not. Well, we don't know. We have to go through it. That's the process of elimination. We don't know where the thing is. That's how we get to it.
Yeah.
We all know the things that did work. Didn't Thomas Edison say he had 10,000 failed or 10,000 failed attempts at the light bulb until, yeah, the other 10,000 are now misinformation, but he had to go through them all, and now we can just turn it on and off. That's how we get to it. And
Yeah.
it is kind of, I don't mean to make light of it, but right, everybody do your part. Hey guys, I had a side effect, not you. It's so ironic that it's like you're getting your PhD with the ruthless pursuit of science that is reproducible in other labs and stands up to peer review research, question everything. I agree. That's why I'm really concerned about these like mass vaccine. Shut up, Martha. And it's like, yo, what are we? What? What? That's what I mean. Like it would be hilarious if it'd be hilarious if there wasn't such like serious effects in society. Like it's still kind of hilarious, but you get what I'm saying? Like I'm not trying to, you get what I'm saying. Backing myself
Yeah.
into a corner now. But
Yeah. Well, and you have to weigh the pros and cons as well, right? So whenever you develop even a research question, right, you look into your population, how many are getting affected, what's the survival rate before you propose your treatment, right? Or before you develop your treatment. Usually it's not the other way around of, well, I don't know anything about the populations, but I'm going to propose a treatment for everybody, right? Usually hone in on, okay, so these are the individuals who need it most. If I were to, let's say, for example, on my thesis, I said 0.05 people, die from this. Well, okay, we'll look into that component of, or what other things might be influencing that. So, survival rate, or sorry, that death rate, where then you look at another population of, point or more than that, like 20% of people that get this will die. Okay, well now let's work on that population. Let's drive to get that number down, right, to like that 0.05, right? Because there's so many different factors that could play a role, and we try and conquer the very the things of it. Okay, if somebody gets this, it's a very unfortunate, no chance. That's like, with some of these aggressive tumors, like brain tumors,
12 to
even for
18 months.
pediatric patients. Yeah, it's very disheartening. And you're in this field, you get to see some of it as well. And especially with these children where you have that reality check of, oh, like these kids, if they get this, if they have this brain tumor, it's people are working. There are great physicians, everybody who are working so hard to help these patients, help these people, and give them a longer life. We're just trying to extend that rate. And so I'm used to seeing that sort of thing, right? Of if somebody gets this, it's very dire. Same thing, there are populations then in COVID, if somebody gets COVID or the flu or something else, right? That could be an extreme case.
Sure.
Okay, but again, it's on an individual level, right? It should be on an individual level. You have different populations that are going to respond differently to different medical situations, right? Or different diseases, right? Same thing with diabetes is a huge problem as well, right? Type 1 is, it's very sad. Same with type 2, right? It's still, that is still in something that people can die from. And so there's people that are working very hard on these different subjects where we do see healthy individuals, right? Like the type 1 is genetic, for an example, right? So same thing. And then with cancer, right? You have these people that are really, not before, right now they say, right, things like lymphoma or breast cancer are more treatable than things like brain tumors, right? Well, that's because people had to work hard to get it there, right? People had to make these discoveries and had to question the research, right? Go through this process. Same thing with the vaccines. There's a population that's sensitive to this virus. We'll help them, focus on them, question the different therapeutic options, right? What is going to be the best, not what's going to be the best or make everybody feel good, right? And what can we do that will, and it might not be this, gene or this gene therapy, right? That right, it's a genetically modified protein, right? That we're giving. It's not. Anyways.
No.
So
You're making a brilliant
yeah.
point, and it's kind of triaging on a population scale, and it's also like the right to experiment. I think it might have been Obama, it might have been Trump, I don't remember, but it was called, I think it was called the right to experiment. It was like end of life. You could kind of try maybe a little more exotic therapies that might be lethal,
Yeah.
but to someone whose body's ravaged by cancer, they might not be too concerned with the lethality of it. To me, that's kind of my whole like logic behind arguing for like the use of like in a clinical setting like psychedelics for suicidal people, because they're suicidal. Like they're clearly, they're, and I feel like I can say that. So, but what that means is that doesn't mean that we mandate it to everyone. That doesn't mean, what it means is you go, hey, here is COVID. Most of y'all are going to be fine. Grandma might not be. Okay. And if she does get it, she might survive. There's a good chance she won't. We have this new thing that overall,
Right.
you do. I mean, 20,000 out of 7 billion. I know it hasn't been, but let's just say 7 billion. Statistically, that's actually not a lot of deaths. So, grandma, like you probably won't get myocarditis. You might, but it looks like COVID will most definitely kill you. So maybe you do this shot and maybe you do get a booster every 6 months. Otherwise, or you might not, or you just want to roll the dice.
Right.
Cool.
But that's your choice, right?
And
Yeah.
that's fine. And that's, you get to choose. Hey, it's not foolproof. It's not as safe as a multivitamin, but unfortunately, you're in a position where you maybe, I have a brain tumor. I'm maybe not in a position to go. I'm going to wait for the safer thing. You have to go, doctor. You might enjoy this. So my uncle Tom Collier, I think, started or graduated Duke Medical School in 1980. Smartest person I've ever met in my life. Halfway through medical school, he was involved in a drunk driving accident and had half of his head crushed. I mean, genuinely crushed in. He was brought to Dartmouth. It was up in New Hampshire. And they said that there's no way he could survive. Zero way. And there was it's not brain cancer, but it is related to the brain. They had been trying these, and I cannot remember the name of it for the life of me. They've been trying this like experimental surgery in like the early 80s. And they literally had like their lab attached to the hospital. They had like pigs and lambs and mice, and they were like, it's not at all foolproof. And the main doctor actually said, like, I won't do this. I think he said something like, this is Frankenstein, like, let him die. But my grandparents were like, he's like 26, 28. They're like, in great shape too, like lifter, runner. And they were like, let's do it. And so the chief of the neurosurgery residency, I think he was the chief resident, Dr. Harbaugh, Harbaugh. He was like, let's do it. Like the parents are giving the consent, because otherwise, at best case, he's going to be a vegetable. He's probably going to die. The hemicraniectomy. And they tried it. And my Uncle Tom was the first person on the planet to survive it. He's in his late 60s now. His quality of life, he's been in a wheelchair for about 10 years. He still went on, and so he got the hemicraniectomy, first ever, and then went back and finished medical school.
Wow.
And then worked at Jackson Memorial in Miami in the transplant unit on the top floor, which covers more than 51% of organ transplants in the Western Hemisphere. Went and worked for the Red Cross. Health got worse over the years. His loving wife, my aunt, takes care of him. Yes, he's in and out of the hospital, but you talk to him, FaceTimed him on Christmas. He's still right there. He's like, Tom, your
Mm.
podcast isn't that great. You think you're hot shit. You know, it's like, but he's still there. That
Oh,
that process, that hemicraniectomy is now the go-to for soldiers at like
wow.
Ramstein Air Force Base that come back from war zones. It's used
Mhm.
all over the world, motorcycle accidents, car crashes, construction accidents. Yes, experimental. No, probably will not survive. But we can give it the old college try and we can do it. And not only did it work, he survived. I got to have an uncle, still do, and it has saved countless lives. It was not mandated. I don't remember why I started that rant. But in general, it's you have to push the boundaries of things. You have to
Mhm.
question. They said it couldn't be done. They're like, it cannot be done.
Mhm.
And here is
Mhm.
this case. And but so my uncle kind of, talking about doctors and how come some doctors are like, Martha, you might not get a job after this. As my uncle Tom said, when I was applying to medical school, he said, hey, don't put these people on pedestals. And I was like, why? And he goes, because I finished #1 in my class at Duke and I had half a fucking brain. So, so shout out Uncle Tom. But, so to
That's
give you
good.
some of his advice, don't put him on pedestals. He literally came back and like had to learn how to walk again, had to learn how to eat, and
That
then went back and crushed medical school.
that's awesome.
It's
That
insane.
is awesome.
It's
Like,
insane.
yeah,
It's
that
insane.
is,
I'll get my mom to send me an article or something. I'll send it to you.
yeah. Oh my
Insane.
goodness.
Insane. And
He is awesome.
but
That's unheard of. That is,
no,
wow.
it's the miracle. It is the miracle. I mean, it is the miracle. Like it was not supposed to happen, but he did come back from it and lived. I mean, I would argue some people would say no, he's in a wheelchair now. I would argue he's still alive and is still with his wife. He's had 40 more years of,
Mhm.
I would argue, yeah, a difficult life, but life.
But like,
And and but I mean, all jokes aside, I do take what he said seriously. He was like, hey, dude, don't. I literally finished it with half a brain. Like I went back and finished #1. So he was like, work hard, study for the MCAT. He's like, they're not infallible. He's like, they're jackasses. So take from that what you will, right? And he was a researcher, blood pathology, like headed up, I think, like the Red Cross on the West Coast, like it
Wow.
insane. But
Mm.
so one, yes, experimental shit can be dangerous. That's what science
Yeah.
is. That's how you move it forward. I mean, the first astronauts to die didn't even die in space. They died in a practice run on Apollo 1. They died
Mm.
at the Space Center because something went wrong. But if you want to use it, you can. And if it works, now this new surgery works. And God knows how many soldiers have now it's just a normal thing. And not normal. Brain's always more dramatic. But now it's just like,
Mhm.
oh, yeah, no, you broke your arm. Yeah, no, we can get that fixed. It used to be you get surgery, you're going to die from
Right.
sepsis. Now it's like, no, you're good.
Right.
You can now have
Yeah.
these horrific brain injuries. And because of my uncle, he survived. And it's
Mhm.
now, so it's not just that he survives, it opens a whole new field of medicine
Right.
that affects
Right.
millions.
Yeah.
So, like, mRNA shots might be the best thing ever. Hats off. Hell yeah.
Mhm.
You cannot force people to get these. You cannot belittle them. And as Dr. Bargov said, you can't, this goes against all medical ethics to say, make
Ethics.
people
Yeah.
feel bad. Just ethics. Sorry
Ethics.
for
Yeah.
that ramble now, but
No, no, that was really, really good. Because so even going back to ethics, right? Everybody, even us as researchers, we have to take research ethics, right? Where we even, you learn about the unfortunate story of Henrietta Lacks, for example, right? So I don't know if you're familiar. So um, yeah, so her cancer cells are still used, but her cancer cells were taken from her without her knowledge, essentially.
oh,
So this was back in the 50s. So this is when they were first making discoveries. And then because of that, from there you had to get patient consent for taking samples from, right, the patient's own own, right, tissues,
yeah.
right, so you have to get consent. And so if we learn anything from this, or if you want to think of this in a positive way, is that we have to evaluate the ethics on this situation. Same thing if with the situation with Henrietta Lacks, if nobody stepped in and was like, hey, this is wrong. You can't just take samples from patients, right, who are in pain and have this tumor. You have to have ethics in place, because there will be those people who, it happens, right? We want to think everybody's good, but there are people out there who, that's the reality of it. So these things are in place because somebody had to step in, right, and make that change to make things better. So right now, right, so before that was kind of whatever, right, that was accepted. So right now, if we're accepting this, and that's why it's important to be those people to step in ethically of, hey, this isn't the way to go, right? Ethically, scientifically, right? So if anything, this is why we need to speak of it or about it in the scientific community, because we should try to make the changes for better. So for better scientifically, for the people who might be affected by it, and as well ethically, right? So make sure that somebody can't just do this in the future, right? Because as Bhargava said, now you can make anything a vaccine in one of your podcasts, right?
Yeah.
Yeah.
There's some more, and don't let me keep you, by the way. I'm, like I said, I don't have any podcasts to do tomorrow, so I know I'm just
okay. I know
you.
I know. I was like, I need to eat dinner, but this is.
Do you want me to wrap it up?
Having so much fun.
Okay,
yeah,
well then.
should.
All right.
I know,
me
but
off my own podcast. Great. Well, okay. Well,
yeah.
I'll finish up the point real quick. Sorry. There's a great book called Space Barons by Christian Davenport, and he talks about the kind of the parallels and the differences between Bezos and Musk. They're different approaches, but how they're both wildly successful and worth multi-hundreds of billions of dollars. But there's a quote I love from Bezos, or not Bezos, excuse me, Musk. I don't remember what the exact quote is. He goes, "We often look at the progression of technology as just a constant, right? Like water eroding a shoreline, right? We just kind of accept that this stuff happens. Your hair grows, right? We just expect the weather is just going to start getting warmer. It's February." He goes, with technology, it moves forward because we relentlessly, by the way, I just remember, I haven't eaten either, so actually I just lost all sympathy. He goes, we have to push I swear, he goes, we push these forward relentlessly, and that's what leads to these little changes. We often can look back and go, oh yeah, remember when we had flip phones and now we got iPhone? No, there's a bunch of little things that you don't see, and a lot of people don't see, and a lot of investors don't see, and a lot of people fail. But some people, like a Steve Jobs or an Elon Musk, go, I think it can fucking happen. I think we can do this. And you push and you push and you push and you push, and then it breaks, and we go, well, now it's working. And what you said about breast cancer is like, well, these therapies are working. They were pushed and pushed and pushed and pushed. And that really makes me think about like social issues and like the ethics of this. We just expect that if I walk outside naked, the cops are going to come arrest me. They're still going to read me my Miranda rights. There was a time when that wasn't the case. So even what you're talking about now with taking your cancer cells, these things, these consent forms, your informed consent, you have to know about the alternative treatments. You have to know, hey, by the way, we do get kickbacks from, like, you need to know that.
You need to, conflicts of interest, all of that. Yes.
No, there was a time when this didn't exist. There was, for most of human history, there wasn't the First Amendment, the very thing you and I are screaming about. Yeah, I'm screaming, you're talking about professionally. I'm screamking about till I get red in the face. Is maybe both of our blood sugars just dropping rapidly. But for most of it, we're just dilute. We look back on this tomorrow, we're like, dude, that was fucking horrible. That was horrible. Just
yeah. I know. Next time we both need to be sitting here with a pizza or something.
Just glucose. Just glucose. We'll have Dr. Bhargava moderate it. But the point is right, these things march forward slowly. And in hindsight, some people hit it out of the park. Post Malone, first mix, like outsold the Beatles. Hey man, all right, I'm jealous, but okay. You have to march forward meticulously, slowly, like a glacier moving through a mountain range over millions of years. And you look back and you go, yeah, you've got this valley. It took a long time to get there. And growth is very difficult. You look at like a bodybuilder, it's very difficult and time consuming, and you don't see it happen in a day. So what are we experiencing now? You more than me, because again, I have a podcast. You're actually in this field. Dr. Bhargava is actually in this field. Dr. McCullough is still a practicing physician. Like, people forget that he still sees patients. I know that because I text him and he says, stop blowing up my phone, Tommy. And I'm like, Dr. McCullough, I need to send you this meme. It's still a practice, and the growth is what we're seeing right now is pain. You, Bhargava, McCullough, and like you said, relatively, it's not the end of the world, but it comes at a price. The sit-ins, however many, 62 years ago this month, like the black sit-ins at diners. Yeah, they're gonna call the cops on you. They're gonna. I lived in South Georgia. Yeah, man, a lot of them are still for that shit. Yeah, they're gonna open up the fire hoses. They're gonna release the dogs. Now we look at that and we say atrocious, disgusting.
Right.
But there was that growth period. It wasn't all Freedom March, MLK, and DC. A lot of it was horrifically won at great cost, something that none of us question. And not only do we not question, we go, great, perfect that happened. No one questions the Nuremberg. We go, great. That, but right now what we're seeing is this sort of this microscopic, you're seeing the growth. You go, oh, it doesn't feel good standing out. It doesn't feel good going, hey guys, this is, I know you're going to call me an anti-vaxxer, but this just isn't adding up. I feel like this goes against the courses I take. And then when even your, not just your peers, but the people you look up to are kind of saying in kind of shrouded answers like, hey, you're not going to have an opportunity in the future. Yeah. Well, hey, that's what moves this stuff forward. And you have a much greater respect for those in the past that did it. But you also understand the burden, not burden, the burden, but the obligation that you have to push it forward. Someone has to do it. Someone has to stand up. So with that, I will release you to go get food. But
you as well.
This, I have your permission. Thank you.
You can leave my podcast.
You can leave my. Thank you for having me on Fowlercast. It's been an honor. I hope I don't get you banned from YouTube. But so with that, Miss Martha Fowler, thank you so much for coming on here. Everybody out there, do the right thing. You know what the right thing is in your heart. People are gonna make fun of you. Just do it. Just do it. Fuck the haters. Just do the right thing. Thank you so much, and I will stop.